In the last video, we looked at the role of cytokines and chemokines in the immune response. There is a further component of the soluble response called complement, which is an evolutionary ancient and conserved system, with forms of complement proteins being found in invertebrates like starfish and mosquitoes. The presence of the complement system was hypothesized in 1889 when Hans Buchner discovered that components of serum were able to kill bacteria directly. In 1895, Jules Bordet demonstrated that there were two components to the blood that contributed to bacterial killing. One that was heat stable and one that was heat labile, meaning it could be destroyed by heating the serum. In 1899, Paul Ehrlich named the heat labile component complement as it complemented the other heat stable aspect of the immune system. It was later discovered that this heat stable component was antibodies, with both Bordet and Ehrlich receiving Nobel Prizes for their work. The complement system is made up of over 30 proteins that are mainly synthesized in the liver as inactive zymogens, which means that they need to be modified or cleaved to become active. The activated proteins are proteases, and the effect of this is that one protein cleaves the next in the sequence, resulting in an activation cascade and amplification of the system. Now, something to bear in mind throughout this video is that the complement system is, by its very nature, pro-inflammatory, so it drives the inflammatory process. Before we dive into the specifics of the complement system, it's important to briefly touch on the nomenclature of the complement system, or how it's named. The main proteins in the complement system are designated by the letter C and a number from 1 to 9. But the order that they appear in the complement cascade is not the order in which they were discovered and named. So, for example, C3 comes before C2 and C3 in the activation sequence, but was discovered after C2 and C3 historically. When complement proteins are cleaved, they produce two fragments designated A and B. The smaller fragment A does not remain bound, and there are often soluble mediators with other immune effects. The bigger fragment is B and remains bound. So it's quite easy to remember which is which. A flows away, B remains bound and is bigger. Immunologists, however, love to confuse people, and there is an exception to every rule. And in the complement system, that is C2, where C2A is the bigger fragment that remains bound, and C2b is the smaller fragment that is unbound. Note as well that the C1 proteins are called C1q, C1r, and C1s, and these are not cleavage products, but distinct proteins. When we write out complement complexes, we always write them in the order that the proteins bind. So as an example, C4b2a would be a molecule of C4b, bound to a molecule of C2a. If a molecule of C3 were to bind to this complex and be cleaved into C3a and C3b, with the C3b fragment remaining bound, we would write it as C4b 2a 3b, as the C3b is the last protein to have bound. Right, now that's out of the way, what is the complement system? The complement system functions to destroy invading pathogens, and it does this through one of three pathways. The classical pathway, which is activated by antibodies, the lectin pathway, which is very similar to the classical pathway, but is activated by bacterial sugars called lectins, and the alternative pathway, which is activated by foreign surfaces. All three pathways converge at the C3 level, and the terminal pathway, which is anything after that C3 level, is identical for all three pathways. The result is the formation of the membrane attack complex, or MAC, which punches holes into the surface of the invading pathogen and disrupts its osmotic balance, causing cell death. In the electron micrograph image here, you can see each of these darker areas is a pore that has been formed in the pathogen surface, in this case the bacterium, by this membrane attack complex. We already mentioned that the complement system can be activated by three different pathways. So now we are going to take a closer look at the complement cascade initiated by the classical, lectin, and alternative pathways. The first pathway to be discovered was the classical pathway, so let's start with that one. The classical pathway is initiated by a molecule called C1q, which during an infection can bind directly to a small number of bacterial surfaces via the acute phase protein C-reactive protein, or CRP, but most commonly by cross-linking the Fc regions of antibodies that have bound to epitopes on the bacterial surface. C1q recruits two molecules of C1r and C1s, which are both serine proteases that become activated in sequence as they bind to C1q. This C1 complex, which is designated C1q R2S2, cleaves the next complement protein in the cascade, which is C4, into C4a, the small fragment that floats away, and C4b, the big fragment that remains bound. C2 is subsequently recruited and cleaved. But remember, this is the odd one out, and the A fragment is the one that remains bound, and the B fragment floats away. The resultant C4b2A complex is another serine protease, which is the C3 convertase for the classical pathway. So named because it can bind and convert C3, which is the central molecule of the complement system, into C3A and C3b. C3A is a potent aflatoxin, and C3b is what we call an opsonin, which is a substance that coats a pathogen to help mark it for phagocytosis by neutrophils or macrophages. The C3 convertase is able to cleave multiple molecules of C3, so the surface of the bacterium becomes covered by this protein, making it very visible to these phagocytic cells. The C4b2A3b complex is also the next protease in the series, this time being the C5 convertase, which unsurprisingly converts C5 into C5a and C5b. C5a is a very potent chemoattractant, particularly for neutrophils, so it helps recruit these cells from the blood to the site of infection. The C5b that is produced covalently binds to the pathogen surface and recruits in sequence one molecule of C6, one molecule of C7, and one molecule of C8. Once this complex has inserted into the cell membrane, the first molecule of C9 is recruited, followed by multiple further C9 molecules, which form the pore of the membrane attack complex that we saw previously. This process happens multiple times across the surface of the pathogen, resulting in the loss of osmotic balance, so the bacterium is no longer able to regulate the flow of water into and out of the cell, and this results in the rupture of the cell. The lectin pathway is almost identical to the classical pathway, except that instead of the C1 complex initiating the cascade, it's kicked off by a protein called mannose binding lectin, MBL, or one of the phyclin family proteins, which bind to sugars that are only found on microbial surfaces. Instead of C1r and C1s, two molecules of MASP1 and MASP2 are the proteases, with MASP standing for MBL or mannose binding lectin associated serine protease. And the remainder of the cascade is identical to the classical pathway. The final pathway is called the alternative pathway, and the initiation of this pathway by foreign surfaces is slightly different. C3 is constantly and spontaneously being hydrolyzed in the plasma to form a short-lived intermediate called C3H2O in a process we call tickover. This tickover releases C3A, and the C3H2O binds to a protein called factor B, cleaving it into two fragments, BA and BB. This C3H2O-BB complex is a fluid phase C3 convertase and cleaves further molecules of C3. If this happens close to a foreign surface, the C3b can covalently bind to the foreign surface and interact with a fresh molecule of factor B, again cleaving it into BA and BB. This C3b-BB is the membrane-bound C3 convertase of the alternative pathway and cleaves another C3 molecule. This is the amplification step of the complement cascade, as during activation, over 2 million molecules of C3b can be produced in just 5 minutes. What is left behind is the rather confusingly named C3b, BB, C3b. You may see this written as C3b2, BB, and this is a C5 convertase of the alternative pathway. From then on, the terminal sequence is the same from C5 to C9. Now, as you can probably imagine, the complement system is capable of huge damage if it becomes dysregulated. So to keep it under control, almost every stage of the cascade has one or more regulatory proteins associated with it. These regulatory proteins are present in much higher concentrations than the complement proteins themselves. On top of this, components of the complement cascade are mainly produced as zymogens, or inactive precursors that need to be activated. And once they are activated, they're extremely labile, with a very short half-life. It is, however, a balancing act. The system needs to be tightly regulated enough not to damage host cells, but not so tightly regulated that it cannot respond to an invading pathogen. A few of the important, but by no means all of the regulatory proteins include C1 inhibitor, which is a protease inhibitor that binds and inactivates C1R, C1s, and also MASP1 and MASP2. Decay accelerating factor, or DAF, which blocks the formation of the alternative pathway C3 convertase and accelerates the dissociation of preformed C3 convertases by displacing BB. Membrane cofactor protein, or MCP, which has cofactor activity, meaning it can clip C3b and inactivate it. Factor H is one of the main fluid phase regulators and has both decay acceleration and cofactor activity, so it can break apart the C3 convertase and inactivate C3b. And finally, protectin prevents C9 from binding to the C678 complex and forming the pore of the membrane attack complex. So as you can see, the complement system can be regulated at many different levels to ensure tight control over its activity. So that's an overview of the complement system, which is the evolutionary conserved system that can help protect us directly against pathogens. It's fundamental that the complement system is tightly regulated to ensure that it does not activate against our own cells, as it is capable of huge damage. So now we've covered most of the main components of the innate immune system. You might be wondering, however, how the immune system knows what type of response to mount. How does it know if the invader is bacterial or viral, fungal or parasitic, and then tailor its response accordingly? In the next video, we will look at pattern recognition receptors that allow the immune system to do just that.